Thanks very much, Andre. I really appreciate the invitation. I'm not traditionally a biophysicist, but I keep finding myself involved in biophysics projects. So I'm really happy to be here to tell you guys about some of the work we've been doing. What I prepared today, since Andre said that usually these are a half an hour long, but since I'm the only speaker, I kind of combined two things. So I'm going to tell you two stories about biology in the air. The first part about hitchhiking microbes is some ongoing work that we're doing in the lab, where we're trying to use acoustic levitation to study how microbes survive in atmospheric environments. But then I'm going to tell you a little bit more complete story that we've submitted for publication on parasitic jumping nematodes. And electrostatic forces. So let's go ahead and get started. And do you usually ask questions during the seminar?
We usually just put questions in the chat and then ask questions at the end.
Okay. I just didn't know if I needed to monitor the chat or something.
No, just keep going.
Okay. Yeah, so when you think about biology in the air, the first thing that might come to your mind is health, right? We're all thinking about COVID, disease transmission, maybe mold, other things that you could find in your house, these kinds of things. But that's not really the type of biology in the air I'm going to talk about, not necessarily in your house. I'm going to be thinking about microbes that could be all over the planet, right? So we know that microbes live in the ocean, extremely important for all kinds of processes. They live in the soil, also extremely important. But there's another kind of biome that we've been thinking a lot about, and that's the atmosphere. So we know that microbes can also live in the atmosphere. They can get trapped on dust and smoke. They get shot up out of the ocean through sea spray, which is a common way that aerosols are generated over the ocean. Then they can get transported by wind. They can come down through precipitation, get trapped in rain droplets. And so they kind of cycle through the atmosphere. And so some of the questions that we're trying to think about and ask ourselves are, is this just a passive way that bacteria move around, meaning that it's not really that important? Or is it kind of crucial that they are able to cross-fertilize different biomes by being transported through the atmosphere? And are they growing in the atmosphere? Are they evolving in the atmosphere? And what does their survival look like? So see, these are some of the questions we've been asking in the lab. And so these kinds of pictures have motivated some of the things that we work on. Here you can see dust from the Sahara Desert being blown across the Atlantic Ocean. And this kind of dust transport actually provides a huge amount of nutrients to the Amazon rainforest. We've known that for a while, but microbes and other things can also be transported on these bioparticulates, so to speak. But we don't really know, we can go up there with an airplane, you can capture them, you can try to study them, but we don't really know what their life is like up there. Here's another image just of the recent wildfires in Southern California. Wildfires are particularly interesting from a aerobiology point of view because the fires are happening in an area which is, abundant with life, abundant with soil microbes, and in the trees as well. And so all kinds of things can get launched into the air through these convective plumes from the soil and other places with wildfire smoke. So trying to understand exactly what survives in plumes like this is kind of important. And one of the projects that I'm not going to be talking about so much is we're partnering with some folks that I'll say at the end, and we're trying to launch balloons into these kinds of plumes and capture soot particles so we can study what is actually growing on them or if things are alive on them. So we know that microbes live on particles and in the atmosphere, everything from bacteria to viruses, pollen, of course, microalgae. So there's lots of different things that we know live up there. I'm going to be talking mostly about bacteria, at least that's what we're interested in right now, which is in the upper left-hand corner. And we're going to kind of narrow down, we're going to keep narrowing down our questions because there's obviously a huge plethora of questions that you could ask about this system. And first, we're just going to ask, how are they surviving up there? So many studies have shown that all different types of bacteria live in the atmosphere. Like I said, often what these studies do is they'll either have some kind of high altitude, school bus size NASA balloon, or they take airplanes up there with a vacuum and literally just sample the air, take it back down to the lab, and they find that roughly 50% of what they find is culturable. Um, and what they find are bacteria from all different types of sources, anthropogenic sources, natural sources, um, everything from E. coli, gram-negative, gram-positive bacteria, all kinds of things live up there. Even ones that you would be shocked that might be able to survive, they bring them down and they're able to culture them in the lab. So they're up there, low pressures, minus 40 degrees Celsius, extremely dry environments. How are they doing this? And so we're going to kind of focus in on drying and desiccation in aerosolized cells. So what can that do? Desiccation generally causes membrane damage, protein denaturization, and DNA damage as well. There have been lots of studies for the last 75 years, as far as we can tell, trying to understand how bacteria survive in desiccated environments. Some studies create aerosols directly and then sample them, a few seconds later. And some studies have just look at drying droplets on surfaces to see how long bacteria can survive in these cases. And they find weird behavior, a non-monotonic survival. I mean, relative humidities seem to be kind of the worst for them, or intermediate relative humidities seem to be the worst for them, whereas they maybe survive better at drier conditions or at wetter conditions. So there's a lot of mystery still going on. And this, like I said, this article here, citation number 1, shows you how long people have been thinking about this problem, how long they've been doing experiments as well. So we've really been focusing on this relative humidity aspect and trying to understand exactly what helps them survive. One of the ways that we're doing this is that we make acoustic levitators in the lab. So this is just a cell phone movie from one of our 3D printed acoustic levitators. We have 10 speakers that are kind of aimed at each other, and they run at 40 kilohertz. You can't hear it, obviously. Well, the sound's turned down, but you wouldn't be able to hear it anyway because it's at 40 kilohertz. And what you're seeing is they're levitating a little tiny aerosolized ball there, and then we turn it off, and the ball falls down into a collection vial, and then we can examine it later. And we can levitate these particles for weeks at a time. And the way we create them usually is we take a little droplet, we put anything in the droplet that we want. Maybe we put some dust and dirt in there. Maybe we put salt. Certainly we put bacteria in there. We let it evaporate and form its own little aerosol. And then we can let it hang out there and in any kind of condition that we want in an environmental chamber, change the temperature, change the humidity, and then examine its survival later. This particular little particle here is maybe about 100 to 200 microns in size. I know it's a little big for an aerosol. We'd love to be able to do a 5-micron particle, but that's kind of difficult. But we're working on even miniaturizing it even more. And just to kind of whet your appetite a little bit, this is a bacterial ball that we made. This is just kind of a dense bacterial suspension that's been evaporated and held in there for a while, and then we put it under the microscope. It looks like a weird shaped donut. That's because the acoustic forces kind of deform it as it evaporates into this shell, but this thing is 100% E. coli, actually. We've been examining 2 different types of bacterial species. So it's almost like a 3-dimensional biofilm. And so that's one of the things that we're trying to understand is how important it is for it to have lots of different neighbors in your survival when you're in this aerosolized state. And so this is ongoing work. We've been doing things a little bit even more simply to get started. We're thinking about desiccation, but before we even stick things in a levitator, we're just trying to examine things on the surface with drying drops. And so we've been doing some fairly straightforward, at least conceptually, experiments where we take bacteria, we put them in a drop on a glass cover slip, we dry them for 24 hours in a humidity controlled chamber. We recover the cells from the glass cover slips and rehydrate them in a buffer. And then we just use serial dilution to measure colony forming units. And we've been measuring E. coli, which is just kind of a, a control strain that we've been using. But Pseudomonas syringae, one of the strains we've been looking at the most, this is a very common plant pathogen and is often found in the atmosphere. If you're familiar with aerobiology at all, you might recognize that Pseudomonas syringae is the strain of bacteria that helps ice nucleate at higher temperatures. So they use it to make commercialized snow. They purify the protein from it and use that to help make fake snow and all kinds of things, which is actually banned in some country. I think it's called Snowmax. But if you're interested, it's a kind of a fascinating bacterial strain. And then we test a range of humidities. And what we found, this is just a picture showing kind of like a serial dilution so we can measure colony forming units. But the important thing is what we found is that if you dry this drop at 85% relative humidity, they survive fine after 24 hours. If you dry it at 10% relative humidity, you have 5 to 6 orders of magnitude less survival rate. And maybe that's not surprising. You're like, well, yeah, I take in a pure water droplet, I put them there. If the water evaporates faster because the humidity's smaller, then yeah, they're going to die faster. No big surprise there. What we did find a surprise is when we started to add salt to the droplets, and I know there's kind of a lot of bars on this graph, but maybe you guys can see my cursor. I'll just emphasize what I want you to see. Basically, we're increasing the percentage of sea salt, and it doesn't make a difference at 85% humidity, but at lower humidities, suddenly you get to 3.5% sea salt, and they survive almost as well as they do at high humidities, even though they are drying a whole lot faster. And so somehow the salt is helping them survive. Maybe it retains water. That's one thing we're thinking. Maybe it prepares them for the stress they're going to feel when they dry. I don't have time to go into the whole story, but we think that there's important effects that happen before the drops even dry on their metabolism and physiology. There's also effects that happen after the drop dries. Okay, I'm going to tell you a little bit about what happens after the drop dries and the patterns they make on the surface. But this is the mystery. So drying at 85% humidity yields high viability regardless of salt. And even at 10% relative humidity, adding salt makes a big difference. And so you can just say, well, what are the patterns? What do these dried drops look like? Here is water and salt. This is kind of a control experiment, a little drop dried on a glass slide. And the water evaporates, the salt nucleates, and makes a nice little salt crystal. Very beautiful video. I never get tired of looking at these. If we do water and bacteria, well, the contact line doesn't really pin at all, and you just get a little kind of evaporated mat of bacteria afterwards. But funny enough, when you put the two together, the contact line remains perfectly pinned, and you kind of get this salt flat that has bacteria kind of built into it. And this turns out to be really, really helpful for their survival. And so again, once you put them together, then the contact line stays pinned and the whole thing evaporates into this kind of salt mat. So the question is, where do the bacteria accumulate? So we've kind of varied the cell density and varied the salt concentration, and having a reasonably high salt concentration with a high optical density of bacteria to begin with creates these salt flats. And one thing that we know that's important is after it dries, there's this extremely long period where water is retained and it takes a long time to get out of the droplet, if it ever makes it out at all. Think about little caverns of salt holding water. And you can kind of see them in this video. So here's a close-up drying video. And you can see these little bubbles, water migration still kind of happening after it's dried. And remember, we're sampling these 24 hours later. They're drying usually within minutes to tens of minutes, things like that. And then we're sampling it 24 hours later. So we can weigh the drops too. I have the data, but I don't have time to show it right now. But there's definitely water retained in these, and we think that's really helping the bacteria. So anywhere the bacteria could fit into little tiny caverns in a bioparticulate and maybe retain water, that's a crucial aspect for their survival. So this is ongoing work. We're trying to now take some of these ideas and then dry salt droplets in the acoustic levitator to compare to the drops dried on a surface. Both are relatively important because raindrops and other things can dry up on surfaces, and then these things can get lofted in the air later on. It's really still kind of a mystery exactly what these bioparticulates are. Are they free floating bacteria at 10,000 feet, just 1 micron size bacteria? Or are they hanging out on pollen and other particulates? This is something we're starting to collect samples on our roof and try to answer some of these questions. So that's the first part of my talk, trying to understand how bacteria survive in the atmosphere. And I just kind of told you a little piece about desiccation and how bacteria are surviving this desiccated state. I'm going to switch gears a little bit now. And instead of a global picture where we're thinking about bacteria, at 10,000 feet, all hanging out all over the globe, we're going to kind of like take it back down to the city level. Here's a picture of Atlanta, where Emory University is. We're actually kind of a few miles north. You can see it we're kind of up on a hill. You can see it, the skyline, some of the taller buildings, but this is more of a picture. If you were in Georgia Tech or something like that, you might be able to take a picture like this. But if you look at this grass down here, we're gonna kind of we're still in the air now, but we're going to come down to the level of the grass and think about kind of what's going on there. And what lives in this area are nematodes. Many of you probably work with nematodes. Usually, the kind of standard model organism that people study: swimming, locomotion, neurons. They do study, the neurons in nematodes and all kinds of other things. But we're thinking about a very particular type of nematode that's related to the model organism, except this one is actually parasitic, and it infects insects. And down in the bottom right, I have a picture of one of these nematodes launching itself into the air, and it actually gets attracted to this fly and through electrostatic forces and infects this and can, well, this is a dead fly in the picture here, but we work with live flies too. And can infect these insects. And so the question is, why do these things jump? What affects their motion while they're in the air? And can electrostatics be important? You might be thinking, why are we thinking about electrostatics? And I'll explain exactly why we know that those are some important forces. So these nematodes are about 400 microns long, about 20 microns in diameter. So these are the dimensions you should keep in your kind of head as I'm moving forward thinking about this jumping nematode here. So here's some videos from our collaborator, Victor, who I'll mention again at the end, so that we know that flying insects are naturally charged due to the beating of their wings. This is a complicated process. It's fluid electrification. Just like when you rub two materials together, you can transfer charge between them. It's called triboelectrification. But we know that things that fly through the air, even two liquids flowing against each other, can transfer charge. And we know, for example, that bumblebees, electrostatics is extremely important to them. It's how they collect pollen, for example. But you can imagine if you're a little nematode and this charge object comes up to you, the question is, can evolution use this fact to help these nematode worms attach to their host? And so in this video here, you see this little nematode jump, flinging itself, and then attaches to this host and will then infect it. These aren't videos from my lab, they're videos from Victor's lab. He's at UC Berkeley. We've been partnering with him on this project, and I'll mention more at the end. So here's kind of a more controlled experiment where a nematode jumps. This fly is connected to a wire and it's charged up with a voltage similar to what you might find in the environment. And this worm looks like it's going to fly away, but then electrostatic forces take over and it's attracted to it, and then boom, it hits the fly. And so we were taking videos like this. We can track the position of the worm in time, and we're trying to understand how can we infer the charge on the worm and other things that determine its trajectory. And so the research question we're asking and trying to answer here is, how much do electrostatics and aerial drift facilitate host attachment? And so these worms, again, they pull themselves down into this latch, and then they fling themselves up and spin in the air, and they're they're oppositely charged to the insect, obviously, because they're attracted to it. If you're interested in this kind of loop formation and jumping, it's a really cool problem. Sunny Kumar in Saad Bamla's lab has a very nice paper on this. You can learn all about it. When you track these jumping worms, you see these are some of the trajectories from 2 different experiments where they were launched at different vertical heights from the fly. And what's important about these is that they're launching themselves at about 1 to 1.5 meters per second. So it's pretty fast, okay? And they're spinning initially, and then eventually drag takes over and they stop spinning and they're attracted to the fly. But that's the velocities we're thinking about here. And just so we want to, we're physicists, so we want to make sure that everything can be approximated as a sphere. So we replaced the fly with a spherical metallic ball and did many experiments with that as well, just as a control experiment. We're also going to be assuming that the worm is a sphere for simplicity in our analysis, but it turns out that it's a pretty darn good approximation. And so we want to have a model for the airborne worm. And essentially, the equation of motion for this worm is using Newton's second law. We're modeling the fly as a single point charge up at some distance away from the surface. And as long as the size of the fly compared to the distance to the surface is small or reasonably small, then this is a good approximation. And so, of course, the surface that the worm is jumping from is grounded, right? It's kind of a wet filter paper that's grounded. And so we have an image charge of the fly beneath the grounded plane, and that's modeled as this blue negative charge down here. And these are the 2 terms in the equation here that represent the electric force. This is just the method of images that many of us learned when we were in undergrads. Now, the worm here is this negative charge, and you might be asking, well, shouldn't the worm have its own image charge as well? And the answer is yes. That is important when the worm is touching the surface, but once the worm jumps off the surface, its own image charge is kind of negligible. So we disregard it from the model because it makes the computation a lot faster. So then we also have Stokes drag that we're using, and there's kind of a free parameter here. There's this hydrodynamic radius, AH, that we're going to try to infer from our experiments, and then there's gravity. So there's 6 unknown parameters in the model. There's the worm charge, there's the hydrodynamic radius, the initial jumping velocities, and the z position of the worm. And we have this 2-dimensional data, but we're going to need to infer any velocities out of the plane as well. So this is the data is 2-dimensional, the actual experiment's 3-dimensional, so we have to infer some unknown things. So in order to do this, to fit all of this data, we use a Markov chain Monte Carlo method to fit the computed trajectories to the experimental data, because it's a lot faster in finding parameters that are good fits than just trying to do some kind of gradient descent or something like that. That gets stuck very easily when you have this many parameters and trying to fit trajectories in 3 dimensions. Okay, so This is just some 3D trajectories showing that the fit is pretty good. So this is a sphere that's at 100 volts, 200 volts, 300 volts, and these are some trajectories of jumping worms and the fits to them. Um, but if you look at the fits in 2 dimensions, they look a little bit better. And we were kind of surprised that it works so well, even for some of these ones that are really weird, where it shoots off to the left. For example, this red one on the left shoots off to the left, makes a almost 180-degree turn, and then comes back to the fly. So the spherical worm and spherical fly model fits the trajectory surprisingly well. And again, we're primarily interested in the charge on the worm and the hydrodynamic radius of the worm. These are the things that, are the most physical parameters that we want to infer. And then we want to ask the question, are electrostatics even necessary for success? Does this fly even, if a fly comes down, does it matter at all if it's charged up for the worm to hit it? And this is going to be a recurring theme. We think the answer is absolutely. For example, you can take those trajectories, you can turn off the electric force, and essentially you get parabolic motion with drag. And only, I think the success rate for, one trajectory here ends up hitting the fly. And so if a worm, if a fly comes down and it encourages the worm to jump because it's beating its wings and the worm can feel this airflow, if it jumps, the chances of success are pretty low unless there's electrostatic forces. And so we can take some videos. Here's a worm taking off into the air, and then drag takes over. Of course, the x component of the velocity gets pushed to 0, and then it just kind of falls straight down. Okay. And so one important question we're trying to understand is, how does this worm get charged? I mean, this is a very humid environment. It's got to be humid for the worm to stay alive. So it's near 100% humidity. Is it being charged in the air? Is it some kind of bioelectricity, or is it just induction? And so we think it's induction and I'll show you why we believe that. The way this might work is this fly beats its wings, becomes charged. This induces polarization in the nematode. If it's connected to a ground somewhere, then you can have a net current go to ground and leave a net charge on the nematode. And then when that nematode jumps into the air, it's going to have a net charge and have a monopolar attraction to that fly. And it doesn't matter if the fly is positively charged or negatively charged, this mechanism should work either way. And so you might be wondering, well, what kind of charge would we expect on this nematode? And so we, dug back into the electrostatics literature throughout the ages, and turns out, of course, that Maxwell solved this problem, 150 years ago or whatever. He solved everything. But it's actually kind of an obscure problem. And so the problem is you take a plane that has a finite charge density on it, or a known electric field that induces a charge density. You bring a sphere from infinity, a conducting sphere, you touch the surface and you lift it off and you ask how much charge is now on the sphere. And the answer is 2/3 pi cubed times the radius of the sphere squared. And I put it here in terms of the electric field, the local electric field, which determines the local charge density on the surface. Okay, and it's actually kind of hard to find this solution. You can compute it through a method of images, but I actually found a thesis back in 1968. Fantastic thesis, by the way, that gives all the details of this. And so this is our theoretical prediction. This is what we think. If it's induction, the fly applies an electric field, induces charge density on the surface. This is what we expect the charge on the worm to be if it's a sphere. And A would be the radius of that sphere. So let's see how good the prediction matches our experimental data. So in this graph here, this is the charge that we infer from our Monte Carlo method, okay, in picocoulombs. This is the local electric field at the position of the worm where it jumps. So maybe if it jumps right under the fly, it has a higher electric field. If it jumps from, kind of over here away from the fly, it's going to have a lower electric field. So we take into account whatever electric field as the position it jumps. The data points are from worms jumping on both real flies and spherical conductors at different voltages. We have a power supply that we apply the voltage. And the green cone here is what you would expect from Maxwell's prediction if the radius, if A, the radius you put in of the spherical worm was 100 microns and 200 microns. So all of our data is kind of contained within this range here, which is pretty good because we know the worm is about 400 microns long, about 20 microns in diameter. It's not a sphere, but if I balled it up like a polymer and squished it together, it might have a radius that's about 100 to 200 microns. So it's pretty consistent. So this kind of shows us that induction is the mechanism going on that's giving charge to these worms. So we have a prediction for what their charge is when they jump. The next question we're going to ask is, okay, well, worms also jump in the wind. And in fact, people know that these nematodes can be not only just jump in the wind and land somewhere else, but they can actually be lofted and carried kilometers away. Okay, and so they're not that small, but they're not that large either. So they can be lofted and drift for quite a long ways. And so in this experiment here, we have some light wind blowing at a less than about a meter a second or less than a meter a second, and it pushes these nematodes in this direction. And we want to ask, can this drifting actually help them achieve their goal of infecting the fly as well? And so these experiments here have no fly at all, no electrostatics. We're just looking at their drift and trying to model it. And it turns out that you have to be a little careful. You have to be a smart fluid mechanics person and know that there's a boundary layer near the surface. And so what we actually use is the Blasius, kind of the classical Blasius solution for the boundary layer near the surface. And this is kind of important because not only does it have an x component of the velocity, but because the divergence of the velocity has to be zero, there's also a small y component of velocity, which helps loft them up a little bit higher. And so by using this kind of known velocity distribution, we can fit the trajectories of the worms and infer a hydrodynamic radius as well. And what's really interesting is we can capture the transition between Stokes drag and quadratic drag. So at low jumping velocities, we infer a hydrodynamic radius, AH, assuming Stokes drag, right? We're inferring this AH, and we get a constant answer among all of our different experiments, around 100 microns, which kind of makes sense. The worms are very uniform in size. They're jumping, and we're inferring this hydrodynamic radius. So at low velocities, it's basically Stokes drag. And then the Reynolds number increases as the jumping velocity increases, and it starts to kind of be more consistent with quadratic drag. And what this means is that quadratic drag should go as the radius squared. So if I'm inferring the hydrodynamic radius, and it goes as u squared. So if I'm inferring the hydrodynamic radius, it's not going to be constant anymore for my inference procedure at higher velocities because the drag is not Stokes drag anymore, it's quadratic drag. And so we can kind of see this transition here. And there's data from both electrostatic experiments here and just these wind experiments here. And so the whole point of this slide was that what I've told you in the last few slides is essentially we think we're doing a really good job of getting the charge on the worm. We're doing a really good job of getting the hydrodynamics of the worm and this hydrodynamic radius. Now we're going to try to put all of that together. And so what the postdoc in my group, Ron, who really did all this work, who I'll advertise at the end, he then took our model, now that we know these parameters, and just simulated it thousands and thousands and thousands of times and look at the capture rate, right? We wanted to know how much electrostatics and wind help these worms jump and reach their target. And so we can just simulate it thousands and thousands of times and look at how often they get captured. If the wind is blowing too hard, it can actually blow them over their target, right? So it's not clear that wind is always a positive thing, especially if you jump from the right side, the wind can just blow you away. Whereas if you jump from the left side, it might help you achieve your goal, right? And so we can simulate this 1,000 times at different voltages, different speeds, and look at the capture rate. And then we can put all that together into sort of a capture probability distribution here. And so this color here is the capture probability. And it's a function of two variables, the voltage on the fly and the wind speed. And as you might expect, having electrostatics is always better, right? You always get a higher capture probability with electrostatics. But what was kind of surprising is there's intermediate wind speed that kind of helps you that helps the nematode reach its host right around 0.2 meters a second. You don't want the wind blowing too hard, but a little wind is always going to be a good thing. And so what we think is going on, the important thing for biology here is that not only do electrostatics facilitate attachment, I think that they could be necessary for the evolutionary strategy of the worm, meaning it's hard to see how jumping would ever evolve as a strategy. That they find hosts other ways. They don't only jump, but if this jumping mechanism evolved, I think that electrostatics had to be present. Otherwise, the success rate is just too low. So it's a significant evolutionary pressure. If you're interested, we've got this posted on the bioarchive. This is a collaboration. Ron did all the work here. It's a collaboration with Saad Balma's lab, and Victor, who was a postdoc in Saad's lab, is now runs his own lab at UC Berkeley. So if you're interested, you should check it out. Pretty happy about how all this worked out. It was a really, really fun project to work on. With that, I'll kind of finish up and thank all the people who have worked on what I've talked about today. For the aerosolized microbes that we're working on, this, like I said, this is an ongoing project. My postdoc is doing all the work. A number of undergraduates have made contributions to this. Min-Soo Kim is a biophysicist here at Emory that I'm partnering with on this project. Josh is a former postdoc in my lab who is working on the balloons, trying to capture bacteria in the air. He's also working with Joe Dufek, who's at University of Oregon. This is a picture of Kieran and Skylar. When we went out and visited, we got to look at their balloons. We even got to launch some balloons. We're there in this picture. They're looking at these solar cells that we attach to these balloons that go and capture microbes in the air. It's a really fun project. Tats, as a postdoc in Min-Soo's group, has been helping us do a lot of the work with the desiccation and imaging them under you saw those beautiful green kind of images of the desiccated drops. That was Tats. And the jumping nematodes, all of those videos, if you saw a jumping worm, it was done in Victor's lab. And this is a partnership with his lab. Again, Rand has been doing all the analysis and the modeling, the inference procedure, the Monte Carlo method, all of that. So it's kind of been a really fun project to work on. Even though we've only had a worm in our lab one time where we actually tried to levitate a worm to see if we could get it to sense electrostatic forces, but that's the only time we've ever had an actual nematode in my lab, at least that we know of. There could be other times, but so with that, I just want to thank everybody you see on this slide here, and thank everybody for joining me today, and I appreciate your attention.